Diversity of Life, Investigation 3.1b, the Elodea plant cell. Last time we were looking at the Elodea plant, that aquatic plant that you'd find in some aquariums and stuff like that, and we did some looking at it through the microscope at very high level power to see what microscopic structures that Elodea plant was made up of. Well, what you saw was it was made up of a bunch of brick-like structures, bricks or boxes, and stacked up like bricks, like on a brick wall. That actually plays an important role in how that plant and all plants are shaped. We'll do some more investigation about those particular things in the plant cell as we go along. The Elodea plant was not the first one to be seen that showed these distinctive box or brick-like structures. Back in the middle of the 17th century, more than 300 years ago, the microscope was a new invention, and Robert Hooke was one of the first people to use to look at plants. He looked at a thin slice of cork. In this case, cork you would use in any kind of cork of a wine bottle, cork of some science labs used to have cork, a lot of things like this, cork wall, like a wall, a board, bulletin board. I'm getting to it here. Anyway, he took a thin slice of cork under the microscope and saw that it was made of hundreds of subdivisions. He thought the subdivisions looked like little rooms. And as you can see over here on this side, yeah, looks like little bitty boxes and rooms. In fact, they reminded him of tiny rooms called cells that monks lived in the monastery. Maybe they didn't have jail cells back then or anything like that, but that's what we might think of today when you think of a cell and a room, would be something like that. In a book that he wrote in 1665, Hooke was the first to describe the tiny units in the plants that he observed as cells. The word caught on, and we still use it today. So all the way back in 1665, we were looking at small structures inside of plants, and he identified them one as cells, and we use that even today. Once again, here's a little picture of the Elodea cells at 400 times magnification. And again, we had you see the different shapes around each of those boxes and how they form little blocks and little bricks and little rooms. And those are what we determined to call cells. The idea that cells were like little rooms meant that the rooms had walls. In fact, that's what we call the borders of plant cells, cell walls. So we're going to go back to part 2 of our Elodea worksheet. And I want you to find the drawing that was in the circle that you label, and label the cell wall on one Elodea cell. So if you have a brick, looks something like this. You're going to draw an arrow to the outside and label it cell wall. The second thing was the actual live version you were looking at, if you were looking at those here in the classroom, was did you notice anything that was moving inside the cells? What did you observe? If you need help with that, there's a section in the FOSS-Web database called Cytoplasmic streaming that shows an actual movement of the materials inside of those Elodea plant cells. Let me show you. Here's what I'm referring to. When you go to fossweb.com, you go to the Diversity of Life unit, obviously. You will go to the multimedia, and then there is a section called database. We're going to be using that for quite a bit of stuff if you haven't done so already. In that one, you're going to find Elodea cytoplasmic streaming, and that will show you a short little video of the particular Elodea plant cell and what seems to be moving, if there is anything moving inside of it. Let me show you that video next. Here's the video of the endoplasmic streaming. You can see little things moving inside of each of those cells. What are those things, and how would you describe them? I'll play them again just so you can see them one more time. Moving around slowly, being able to make your observations and good, accurate descriptions as we go along. So what did you notice with things inside? Well, there was two things that we're going to focus on. One of them was really pretty obvious, the green balls that you saw kind of floating around. But the more important thing in there is that what they are floating in. Cells are filled with a fluid called cytoplasm. Cytoplasm, if you were to equate like a plant cell, it's got a hard, rigid wall that we're able to see through, kind of. I liken it to maybe like an aquarium. An aquarium has fish and plants and rocks and soil, and the most important thing is water. A cell has the fluid in it called cytoplasm. That is kind of like what the water is. Cells are filled with a fluid called cytoplasm. It is a substance in which all the other parts of the cells are located. Cytoplasm can sometimes be seen moving inside the cells of living plants. And so if you looked really closely at that, you might have been able to see some of the cytoplasm moving around as well as those green balls. So the next thing is go back to part 2 of your worksheet and label the cytoplasm in one of the Elodea cells. Remember, you had an Elodea cell look like this, and you put an arrow to the outside for cell wall. So what you need to do next is some part inside of it that is not a green dot, you're going to label cytoplasm. Probably the easiest thing to see in there was the little balls, green. I should put up there, green color, balls moving in the cytoplasm. And these are called chloroplasts. Chloroplasts, they contain a pigment called chlorophyll that gives plants their green color, and it does an important job in that it captures light energy for the plant. So the green that you see in the plants is actually this pigment called chlorophyll. If you run and slide on the grass and you get a green stain, that stain is this chlorophyll that is staining up your pants or socks or shoes or whatever it is. If you mow the lawn, push mowing it, and you wear white sneakers and you follow behind the mower, and as you're walking through it, eventually you'll get green on your shoes. That's because as the mower is cut and released some of that chlorophyll, it builds up on your shoes and makes them green. So those green balls we call are chloroplasts because they contain the chlorophyll. So the next thing I want you to do is in your drawing for part 2, go ahead and label chloroplasts in one of the cells. We did cell wall, we did a cell, the cytoplasm, so you're going to draw an arrow to one of those green dots right there, and chloroplasts, H-L-O-R-O, plasts. For those, that's great. Once you've finished that, you need to go ahead and upload that or submit that worksheet on the Elodea plant. Go ahead and turn that worksheet into Canvas, and that will do your work for 3.1a and 3.1b. Next, we'll focus on more on what else is inside of the plant cells besides those things. Thanks.